Hello, everybody. Welcome to the WMIS IDS webinar. We have a pleasure of having today Dr. Thomas Janquilo from UT Austin. The title of his talk is, hello, everybody. Welcome to the WMIS IDS webinar. The title of his talk is Toward Practical Computational Framework for Personalized Clinical Trials. I'll start with some few updates from WMIS. So as many of you know, we have the upcoming annual meeting in Prague. The late-breaking abstract submission is open, so please submit your abstracts. We also have a webinar tomorrow on gene reporter in vivo tracking by the iState interest group, so please register if you haven't already. For those of you who are members of IDS and those of you who are not, we have an open position for vice chair. The new vice chair will take over. We'll start this annual meeting in Prague, and will take over as chair in 2024. The mission statement of the interest group is outlined here. Broadly, it's to advance application of image data science across all domains of interest to WMIS. We're focusing on quantitative imaging, radiomics, computational imaging pipelines, imaging physics, QA/QC, machine learning, integration of image data science with multi-scale information, and relevant to today's talk, development of computational models of human disease in concert with imaging and data, and of course, imaging informatics. You can't click this link, but if you go to the WMIS website and sign up to the interest group, we would love you to have join us. And again, if you're not members, join in. And we are, again, like I said, we're looking for a new vice chair that's going to be open to only IDS members. So please join in the group and apply if you're interested. The WMIS will send out the announcement for the vice chair position later on and how to apply to the position. And with that, I'm going to just introduce Tom. Tom received his MA in Applied Mathematics and his master's in physics from Indiana University, and his PhD in biomedical engineering at Stony Brooks. He completed the postdoc at Vanderbilt and climbed the ranks to full professor in 2010. Then he joined UT Austin in 2016, where now he's the Moncrief Chair of Computational Oncology and Professor of Biomedical Engineering, Diagnostic Medicine, and Oncology. He's a founding director of the Center for Computational Oncology and also served as the Director Center for Imaging Research within the Livestrong Cancer Institute at UT Austin. He's the adjunct professor in imaging physics at MD Anderson Cancer Center. And overall, his goal is to develop forecasting models for cancer, integrating imaging and mathematical biology. And on top of all this, he's a great guy, and I think we're up for we are up for an interesting and engaging webinar. And with that, Tom, I want to hand it to you. I want to stop sharing my screen. Let's see. Go ahead, Tom.
All right. Can you confirm that you can see and hear me?
Yeah, we can see it.
All right. Well, here we go. Does everybody know how to ask questions if they have any questions?
I think you probably should have mentioned that. Please put your questions in the Q&A and on the society website, on the system. There's a Q&A tab. Just enter it there, and then we'll read it up toward the end. Thank you all.
Okay, cool. All right. Well, thank you so much for this opportunity. I hope this will be a lot of fun and entertaining, and maybe even learn something. All right, so I'm going to start in left field here. So in the 1940s, the U.S. Air Force tried to make a seat to fit the average pilot for its first generation fighter aircraft. And there was all these crashes that were happening. And after much consternation, they came to the conclusion that it really wasn't the engineering. There was something else at play here. So yeah, they hired this young scientist who had just recently graduated from Harvard, and he was sort of obsessed with measuring body parts. Like his senior thesis at Harvard was to measure the size of all all the people's hands there and see what the averages and what the distributions were. And so they hired him to measure 140 different dimensions on more than 4,000 pilots and potential pilots. And they focused on what they thought were going to be the top 10 dimensions that they thought were going to be the most important to see for making these cockpits comfortable. And so then they tried to find the pilots that were within the average of these 10 different dimensions. And average was described very liberally here. It was the mean plus or minus 30%. So they're trying to find the pilots that fit into this range. And so of the 4,000 pilots, this is where I would normally ask for audience participation and say, how many pilots do you think actually fit into this average 10-dimensional space here of these 4,000 pilots? I'll ask you to pick a number in your head, and whatever that number is, I doubt it was 0. But so 0 actually fell within this average 10-dimensional space. And so this guy, this Gilbert Daniels guy says, you know, if you design the cockpit to fit the average pilot, you're designing it to fit no one. And he said, once we showed them that the average pilot was a useless concept, they were able to focus on fitting the cockpit to the individual pilot. And obviously this isn't one of my area, this isn't an area I have much expertise in, but I've read that some of the initial things on how to make these seats comfortable are actually things that found their way into all the adjustable parts that are in your car seat to try to make you comfortable when you drive around. So nowadays, all cockpits need to fit the pilots whose measurements are within 5 to 95% range of each of whatever the important dimensions are determined. All right, so a similar problem exists, of course, for clinical trials, right? The fundamental issue of clinical trials, as I see it in my infinite wisdom, is that clinical trials identify the intervention that works for the population. Of course, there's no average person. So by finding the therapeutic regimen that works best for everyone, you are in fact finding the therapeutic regimen that's going to work the best for no one. This is being overly dramatic, but there's still a lot of truth here. You know, humans are very heterogeneous. That's why this disease is so horrible. It's because it's so heterogeneous from person to person. So if you're trying to land on a therapeutic regimen that's going to work for the population, and there really is no average person out there, then it's just not going to work the best for anyone. Now, of course, statistical inference, of course, it's enormously powerful, but it does rely on properties of large populations that can obscure conditions that are specific to the individual. So these types of high consequence decisions, like those that are in ecology, we have to go beyond more than just data analytics. So these decisions must incorporate whatever the important underlying biophysical processes are, so that you can use patient-specific information to calibrate your model that captures those biophysical processes to then make patient-specific predictions. So I'm sure you're on the edge of your seat. You're thinking, Tom, big guy. That's at least how I'm imagining it in my head, that you're saying, Tom, big guy. How could we possibly do this? Well, there are hints out there to how this could happen. So if you're familiar with the I-SPY trial for breast cancer in the neoadjuvant setting, they have this adaptive clinical trial design where they're trying to increase the trial efficiency by minimizing the number of participants and time required to evaluate a new therapy. So it's been around for quite some time now. And so just a 10,000-foot view is, instead of trying to do a standard of care versus one interventional, they'll have a standard of care versus a whole suite of, could be 10 different interventions that they're going to look at in parallel rather than in serial. And so what happens is when a new participant comes in, they have some set of characteristics, and they're randomized to either the experimental or the control arm, and they'll go on their treatment, and then they'll do the patient outcome either by looking at residual cancer burden or pathological complete response. And then you update the predictive abilities of your model, and then that's the model that's loading the patient into the experimental or the control arm in the first place. And then this cycles around. So as you get more and more patients with more and more similarities, then they're more and more likely to go on one particular arm of the trial. And if that turns out well, you can either graduate the thing to FDA approval, or if it's not working, then you'll then that arm of the trial is removed. And so based on what their outcome was, you're going to update the randomization probability so that the next person that comes in, they'll have a higher probability of going in that arm that worked well for the previous set of patients, or if it didn't work well for the previous set of patients, there'll be less likelihood of them to go into that arm. So patient characteristics and outcomes are used to update how future patients are assigned to study arms. And this has actually turned out to work pretty well. So the adaptive design seeks to move patients from arms less likely to help them to arms more likely to help them, and do so as soon as possible in the life of the trial. So what we and others are trying to do is to seek methods to accomplish this for the individual through prediction rather than through the population, which has to rely on assessment. So we're trying to go from assessing things that are happening at the population level to predicting what's happening at the individual level. So ISFI seeks to assess early in therapy which population is going to benefit from a particular intervention. So we and others are trying to predict early in therapy which individual is going to benefit from a particular treatment. But of course, it's not just about predicting a response. Your predictions have to be actionable. So here's the hypothesis, or here's what we're positing. That you can achieve this through the suite of mathematical techniques that have been maturing over the years. One is mechanism-based models. So mathematical models that have some aspect of the relevant biology and physics baked into them. Then you can use these things to power digital twins, from which you can then apply optimal control theory on. And then of course, since it's an iterative process and you don't just do one intervention on the patient, you have to acquire more data and then update your subsequent predictions. You then need to assimilate more and more data as it comes on. So these are kind of the 4 little vignettes we'll go through for the rest of this session today. And I'll put it in all in one sort of grant speak. Mechanism-based mathematical models can enable digital twins to identify optimal treatment plans that can then be updated for the individual patient as they go through their treatment. So hopefully at this point, you are really locked in and excited. And you're excited about the next 30 minutes of your life here. We got to get to about slide 45, I think is where we are here. And a lot of those slides are spacer slides, so don't worry about them. So that's, if you're wondering where we are in the performance, then that's where we are. So we're going to go through each one of these 4 things a little bit, incorporating patient-specific data into these mechanism-based models. And that's where we'll start right there. All right, so if you open up chapter 1 of any book on mathematical biology or mathematical oncology, this is what they start with: exponential growth. And I certainly don't mean to insult anybody's intelligence. We're just getting it going here. So n is the number of tumor cells at time t, and dn by dt, of course, is the rate of change of n, and it's equal to the number of tumor cells that happen to exist there at that time times the rate at which they're proliferating. And you separate the variables, you can solve this in your sleep. And as time goes on, you get exponential growth. So this is what you see for cells in a petri dish at an early time point with plenty of glucose, or even at early time points for cells injected into the back of a mouse. But that, of course, cannot continue indefinitely. You reach this carrying capacity. So if we let theta be the carrying capacity, then we augment this exponential growth by slapping on this logistic term here. So n over theta. So as the number of tumor cells gets close to the carrying capacity, this number gets close to 1. So 1 minus that ratio that's close to 1 is a number that then is close to 0. And so then the rate of change goes to 0 and you level things off here. So this is what you see for later times for cells in a dish as they're proliferating up to the carrying capacity of that dish. As they can know, if you imagine a monolayer growing in the dish, they can't go past the walls and they're not growing on top of each other so very much. And so you get a carrying capacity effect there. Same thing actually happens for tumors growing on a hind limb of a mouse or inside an organ. So these models don't describe any spatial variations in how the tumor is growing. So the next thing we have to do is put in a spatial term, and the next thing to do, now we're like into chapter 2 in that textbook, is to put in a diffusion term. And so now we're working with a partial differential equation. The rate of change of the number of tumor cells at positions x, y, and z at time t. The change of that with respect to time is equal to, again, your logistic growth from up here, and then your diffusion term. And so, of course, if you have 1 derivative in time and 2 derivatives in space, that's describing a diffusion property. And of course, tumor cells don't grow out isotropically. They don't migrate out isotropically. The tumor doesn't grow in all directions equally. So what we do is we actually exponentially damp this diffusion term here by the mechanical properties of the surrounding tissue. I'm not going to go into that today for time reasons, but if you want to talk about that, totally happy to. So here is the repeat of the model before. Tumors don't expand, maybe I am going to go into this. Okay, tumors don't expand in a radially symmetric manner. What we do is we couple it to the surrounding mechanical properties. Okay, so we're all going to talk about it. So here's this D right here. It's the same D that's up here. And we mechanically couple it to, we exponentially damp the rate at which the tumor is expanding by this thing called the von Mises stress. So it comes from this differential equation here. The divergence of the Cauchy stress tensor is proportional, that's that lambda there, to the gradient in tumor cell number there. So you can solve this differential equation for sigma, and then the von Mises stress is just a way to turn the Cauchy stress tensor into a scalar that we can slam into the argument of this exponential to damp the rate at which the tumor cells are diffusing out sort of in bulk. And so in regions where there's higher stress, they're not going to diffuse as far per unit time than in regions where there's less stress. And so this actually does a pretty good job of capturing how these tumors are growing out into space. Okay, The last thing to do for our model is to include the effects of therapy. And so here's the bulk of our model, our movement, our proliferation, and then we have our treatment term over here. So again, over here, this is the rate of change of the number of tumor cells that possess X times T with respect to time. They're growing out into space in a way that's inhibited by the mechanical properties. They're proliferating in a way that's inhibited by the carrying capacity, and then they're going to get some treatment here. And so here we have the N treatments that are given to the patient. Alpha is indexed by N, the number of treatments. It's going to be exponentially decaying as the body removes it from the system, and then we have an estimate of how the drug is being distributed to each position X at time T. And so the question becomes, what do we do with this equation? And so we had this idea almost 10 years ago now for trying to steal a bunch of ideas from meteorology. So just as satellites provide the data for weather forecasting, quantitative imaging data can provide the data for tumor forecasting. I'll just say that if you get nothing else out of this spiel, which I understand is totally likely, and I apologize for that, you might just look up this little thing. It's a perspective, it's easy to read, but it kind of summarizes our whole sort of viewpoint on this. So the idea is that, you can have a global map of, I believe this is a global map of humidity, but you'd also have temperature and pressure gradients and these types of things going into typically the Navier-Stokes equations to then make a weather forecast. We want to do the same thing for quantitative imaging. And so today I'll talk about using two flavors, right? Contrast enhanced MRI and diffusion weighted MRI. So here is, you're looking at a sagittal cross section of a woman with an invasive ductal carcinoma. A nipple is here, chest wall is over here. This is a lung over here. Heart is creeping into the field of view down here. And so this is a patient that was a responder. So after one cycle of neoadjuvant therapy, not a whole lot of change in tumor size, but a good deal of change in our perfusion metric right here, as opposed to a non-responder where after one cycle, not a whole lot of change in the size, but clearly indicating a higher red and yellow pixels indicating higher flow. So if you do the ROC analysis on these patients, your area under the curve is 0.77 for using a quantitative DCE to predict response after a cycle of therapy. And this is kind of old news now, and it's not a phenomenal AUC, but it's not bad. And people are use this kind of stuff now. And then the diffusion cycle, diffusion MRI, it's the same kind of idea. Before therapy, after one cycle, an increase in the ADC right here, indicating a decrease in cellularity, and the AUC is about a 0.81. And lots of people have done these kinds of studies, and these numbers seem to keep popping up. In our own hands, when we combine the DCE with the diffusion, our ability to predict a pathological complete response after the first cycle of neoadjuvant therapy is about 88% accuracy. So that's pretty good. We need to keep that number in our mind going forward here. But what it does here is it gives us some motivation for using these measurements as inputs into a mechanism-based model. So if these measurements on their own, at least statistically, can separate responders from non-responders, then they might be something we want to build into a mechanism-based model. And that's going to be what we're trying to do here. So back to our little metaphor here with our weather model. Global map of humidity along with pressure gradients and temperatures and so forth going into your weather model to make a weather forecast. Now we have these measurements of perfusion and cell density going into a tumor model to make a tumor forecast. So here's back to our model right here that we saw before. There's our movement term, our proliferation term, and our treatment term. And what we're going to do is we're going to take a series of measurements. We're going to make our DCE and diffusion measurements at baseline, and then early in the course of therapy. It could be after one cycle, two cycles, a midpoint, whatever. We use those two measurements to calibrate the parameters in here. So our N values right here, the number of tumor cells at position X and T, that's going to be estimated to first approximation by our diffusion data. Our carrying capacity is easy to assign because we know the voxel dimensions and we have an estimate of the distribution of cell sizes. So we know the maximum number of cells that we can physically cram into a pixel, a voxel. And then our next sort of first order approximation is this, the drug concentration right here. We're going to get that estimate from our DCE data. So to first approximation, where the contrastation gets is going to tell us to first approximation how our drug is going to get there. And that actually works pretty well for the small molecule chemotherapies, perhaps less so with the targeted antibodies that you might see for like HER2 positive cancer or whatever. So here is here's a little, I don't know, just a little video here of overall cellularity. So this is time on this axis here, 3 time points, 2 to calibrate and then 1 to predict. And the red here is supposed to be just a schematic for how these cellularity is going up and down over time. This is the measurements that we make that you have from your diffusion data over time. This is our first order approximation of cell density from our diffusion data. And then as I push go here, this is going to scroll over here and give predictions for what we actually see. And so here's the spatially resolved drug kinetics from the DCE corresponding to the prediction for what the cell density is going to be as it grows over time. And then you want to compare your prediction for to what you actually measured. And I'm going to go over that one more time because that video took way too long to make for just that amount of time. Here's your spatially resolved drug kinetics from your DCE. Here's your estimate of cellularity from your diffusion. And you want this thing to be being compared to what is actually measured. So if we do that over a bunch of patients, here's the observed in red, the predicted in blue, zoom in on that, and just the tumor size is matching pretty good. And you can see that it's not spherical. So that a diffusion term in our model where we couple it to the mechanics around the tissue, around the tumor, that's super important because it gives you this non-isotropic growth. And in fact, before we incorporated that, the tumors that we were simulating were way bigger than what we were actually seeing, which in hindsight makes a ton of sense. But that's what motivated us to include that mechanics. So here's the measured cell counts from your diffusion data, and here's our predicted cell counts from the model. And then if you do the concordance correlation coefficient, it's really super high for these 139 patients from this was from the MD Anderson breast cancer moonshot program. And there's a pretty good match here also for the volume measurement and the predicted volume as well. And so the area under the curve here for predicting path CR versus non-path CR residual disease is 0.89. Now, to those of you at the home audience who have been really locked in and paying attention, you'll say that's 0.89. That is not really any better than what you showed us for the statistical stuff for looking at just the DC and the diffusion, which was 0.88, and this is 0.89, and that's identical. So what's the big deal here? You dragged us through all this math, and we're not getting anything out of it. Okay, so this is where I say we have to take a deep breath and right for the statistical stuff that we talked about before. That was great for doing correlations and separating things on a population. Here, what we did was we calibrated our model with patient-specific information to make a patient-specific prediction about how their outcome was going to be. But we didn't do just that. We also had the prediction for how their tumor was changing in space and time. And if you have a model that's predicting, that can accurately predict how a model is changing in space and time, then you could use that model to go on the computer and try a bunch of therapeutic regimens to see which one might be the best one for that individual patient. And you can't do that with a statistical model, because if you're going to do it with a statistical model, you have to have a gazillion patients with similar characteristics to Miss Jane Doe and have all the possible different therapeutic regimens that Miss Jane Doe could possibly have. All right, so now the next thing I say at this point might ruffle some feathers, and I don't mean to be obnoxious. It's just something naturally that I do. So I really hope that it doesn't come across as obnoxious. But this is why I think trying to take only the AI and big data approach to trying to predict and optimize therapeutic regimens for patients is fundamentally limited. Because if you're going to build, for example, a neural network, which is the current shiny object in the world of AI, if we're trying to build a neural network that's going to select the optimal therapeutic regimen for Miss Jane Doe, then our training data set has to have in it hundreds, perhaps thousands of patients that are similar to Miss Jane Doe. That's already hard to do. And then within that training data set, have all the possible different therapeutic regimens that Miss Jane Doe could possibly get. That's super hard to do because the treatments are getting more and more specific because the disease is getting more and more specific. You know, Miss Jane Doe just doesn't get cancer. She just doesn't get breast cancer. She just doesn't get triple negative breast cancer. She gets one of the 5 or 6 current subtypes of triple negative breast cancer that we know about. So trying to build that training data set that matches what Miss Jane Doe's situation is, that's a really tall order. And I just don't think that's possible. Also, how you're going to deliver the drug in that case is also not sorted out without a mechanism-based model. All right. So we feel like we're getting pretty good at predicting the spatial and temporal development of these breast tumors, at least in the neoadjuvant setting. And so now we feel like we can power them, use them to power digital twins. And maybe I should pause here and say, are there any questions or comments or shouts of joy or screams of misery that need to be acknowledged?
Tom, I don't see any questions in the Q&A. I think you can go on.
Okay, all right. So then we'll use our mechanism-based models to try to power these digital twins. And actually, this is the second thing. If you get nothing else out of this, this is kind of a cool conception for digital twins, and I had nothing to do with it. So you know it's good. So this is an effort that's led by Karen Wilcox, who's the director of our Institute for Computational Engineering and Science here at UT Austin. And she's actually she's an aeronautical engineer. So all of her examples come from from planes and jets. All right, so this is a mathematical abstraction of a digital twin and its associated physical asset. That's what they call their stuffed asset. And there's really two components to this. There's the physical asset, the stuff that's in blue here that you can touch and feel and measure and poke and prod. So they have the control inputs, the actions you can actually do to the system. So for them, things like in-flight maneuvers, sensor installation, and so forth. Then there's the physical state, like what the situation is actually, with the skin thickness, the crack length, how long it extends within the wing, for example. Then there's the measurements you can make. You can put a measured strain or accelerometer data. You can look at the flight logs, all those kinds of things. These are all the things that exist, at least in some sense in the physical world. Then there's the digital manifestations of those things, the digital asset. There's your digital state. These are the parameters that define the computational model. So things like the geometry, the boundary conditions, and so forth. There's the rewards, was the mission success? What was the fuel burn? What's the maintenance or inspection costs? And then there's the quantities of interest, what's the remaining useful life for the patient, and so forth. So these are the two components of the digital twin system. And of course, they speak to each other. But this is a really powerful framework to attack problems in oncology. So here's an example of how this might play out in the case of breast cancer, right? Here, so you have the physical state. The patient presents with whatever their anatomy and morphology is and whatever their physiological and mechanical state is. You can take a bunch of measurements, right? We work a lot with imaging, of course, and so we take a lot of imaging data, but it could be anything else, any kind of measurements you want. And incidentally, these things are indexed by eye to indicate that there's going to be a looping around. It's going to be a recursive process. Then you have your control inputs. You can order more imaging studies. You can order a biopsy. You can try to change the treatment or whatever. These are all the things you can do to your system. And then there's the digital state, the finite element mesh, the boundary conditions, how you're going to give the treatment, for example. So over here on the left-hand panel, you see these little bolus injections, right? The top one is what's done typically for this particular therapeutic regimen. You give one injection, you wait 3 weeks later, and then the next one will follow. But everybody knows that's not the best way to give the therapy to each patient, but that was what was sorted out in clinical trials, and there are not enough patients and resources to run all the trials you might want to run to figure out the best way to give this amount of drug to the patient. Of course, if you have a mathematical model that can accurately describe how the tumor is growing in space and time, then you can go on the computer and try different therapeutic regimens for as long as you can pay the electric bill and to land on the thing that's best. So this is just indicating, all the drug at once, or to give it at 3 different times once a week for 3 weeks, or give it once a day for 21 days like you might do for radiation therapy. All right, so then you have your quantities of interest, the tumor size, shape, cell density, and then of course, with this mathematical model and with this digital twin build, you can try a whole bunch of different doses and scheduling on one patient. And your outcome can be, in this case, we had an outcome that was balanced. There was a balance of efficacy, trying to shrink the tumor while maintaining low toxicity. And so with this mathematical model, you can now build these surfaces. These surfaces are patient specific. They're initialized with patient specific data. You have a whole scheme, a set of doses you might pick, and a whole set of schedules you might pick. And then you can build this thing. And for this particular patient, the distance from the optimal regimen was actually quite far from the regimen that they received, at least according to the mathematical model. But this is, we're right on the cusp. The field is right on the cusp of trying to bridge the gap between making these predictions and actually testing them in prospective clinical trials. It's really exciting to be able to design a clinical trial with a mathematical model driving it. Okay, so then the arrows, the patients come in, they have a physical state, you make your measurements, you build your digital state, you track your quantities of interest and your reward, overall survival, progression-free survival, whatever it is, and then you go back in and you intervene to try to optimize these things, and the whole thing cycles back around. I'm going to zoom in on this thing in the red box here. And so I cut it out, but we have this fluid mechanics model that's supposed to describe how drugs get into the patient. And we want to use that model not just to make predictions, but to optimize the outcome. So everybody knows that the way that we give drugs isn't the best one for each patient. So how do you figure it out? So here's a simulation of how giving one drug to the tumors right down here. You give one bolus injection, and so there's an initial very high concentration in the tumor, but then it wanes very rapidly. Whereas if you were to give multiple injections to this tumor, you can pulse it, and the tumor is bathed in the drug for a longer period of time, even though it's a total same total dose. And that's of course because well, for lack of a better phrase, the pipes for this patient are different than the pipes for another patient. And so the optimal injection scheme for this patient is going to be different than everybody else. And of course, this lets you try a whole bunch of things to try to balance efficacy and safety. Right, so if I pull out some timestamps from those two videos, the top row right here was the video on the left, and this row right here, the one underneath it, is the one from the video on the right. And it says concentration in this little rectangular box here. For the high injection, you get a high dose with a lot of concentration early on, but then it wanes rapidly on the time scale of days here. Whereas if you're giving multiple injections, in this case, it's the other extreme, it's once every day for 3 weeks, then the concentration that this tumor is bathed in is higher per unit time over the course of the regimen. And then that translates into almost an additional 60% reduction in tumor size. So we did a sort of a poor man's version of this. This is 11 triple negative breast cancer patients, just sort of proof of principle. And the blue right here is what the patient actually received. So this was what the patient actually experienced, the percent change in their tumor volume. And then the red one is all simulated using the nonsense I've been telling you about. Is it possible to come with a different therapeutic regimen to have better tumor control? And by different therapeutic regimen, I mean same total drug and same drugs for this. It's triple negative breast cancer, so it's adriamycin, Cytoxan for the first regimen of the treatment there. Just giving the treatment in a different timing. And so sometimes, there's no change, there's very little change at all between what they received in our optimal one, and here there's zero change, they're laying on right on top of each other. But other times there's a pretty big change. This is stable disease to partial response, same here to here. This is progressive disease to stable disease. So on the average, we were able to, at least in principle, land on about an additional 21% shrinkage in the tumor volume than what the patient actually received from using the standard regimen. So this is a hypothesis to be tested. The hypothesis generated from this mathematical model that seems to be doing a pretty good job of predicting who's going to be path CR or not. All right, so that's the bulk of the stuff. The last few things here are sort of where we're going. So I hope you're still tracking with me, or I hope you're still interested. If not, I apologize. All right, so super duper fast introduction to optimal control theory. OCT seeks to determine the specific factors controlling a system. For us, how to treat a tumor so that a specific criteria can be achieved, for us maximizing tumor control. And so how this is formulated sort of just formally here is the rate of change of some parameter of interest. For us, say, take it to be the tumor volume, T, capital T, the rate at which it's changing with respect to time, is some function of that important parameter T, then how you're giving the treatment over time. So a basic example is to identify a treatment plan, call it Z, such that this T of T is minimized at your final time point, T sub F. So we actually had some study that we did many years ago. We were trying to now guide intervention with this OCT. So we had treated HER2 positive breast tumors in mice with Herceptin and doxorubicin. So your targeted therapy and your chemotherapy. And we observed, just sort of by luck, that Herceptin prior to doxorubicin improved the overall tumor control. And so here's some examples of this, right? So here's the control right here. It's this purpley line right here. The tumor just explodes exponentially, a tumor volume versus time here. The doxorubicin, really no difference between that and the control. So the tumor is also exploding there. And Herceptin has a fair amount of tumor control right there. If we combine the doxorubicin and Herceptin together at the same time, we get really good tumor control. The tumor melts away. So that's key to keep in mind. But then if we do the dual agent, this teal one right here with dox first and then Herceptin, it's no different than the control. So that's key. If you put them together, you get good tumor control. If you do dox first and then Herceptin, then you don't have any control. It's just any tumor control, and it's just like the control that got saline. But if you flip the order and you put Herceptin first and then dox, you get fantastic tumor control. And in fact, you can do this with half the dox that was used if you put them in conjugation. So the take-home message there is HER2 positive breast tumors, at least in this tumor line, if we give dox and Herceptin together at the same time, we get good tumor control. If we put dox first and then Herceptin, it's like we didn't give anything, and it's the same as the control. But if we put Herceptin first and then dox, we get the same as a simultaneous treatment, but with half the dox dose. And so that's fantastic because it's those broad spectrum chemotherapies that have all the side effects that you're trying to stay away from. So this is a natural thing to try to optimize. So we developed this model that tries to fit those 6 groups we just talked about, and to build a digital twin for how Herceptin and dox should be optimally delivered for HER2 positive breast cancer. So there's 2 questions that we're trying to go after. using optimal control theory on our digital twin, we want to be able to find a therapeutic regimen that can result in a greater and earlier tumor control. So this I don't know what color this is. I don't know, magenta. This is the thing that we actually experimentally observed. Building our mathematical model, throwing optimal control theory at it, gives us a different therapeutic model that's going to predict that our control can happen much earlier, and then it maintains it. That's one thing to go after. The other thing is, can you have equivalent tumor control with a much less dox? And actually, we landed on about a 60% reduction of dox here, and that's this green line, which is tracking what we had experimentally, but with only 43% of the dox dose. So this makes two very specific parameters, that very specific hypotheses that are testable. And actually, we have these experiments running right now with our collaborators at Alabama Birmingham. So we're all sort of holding our breath to see how these goes. You know, it could be a total disaster, or it could be okay. You know, more than likely it's somewhere in between. But this is the first time that we've actually been able to use our math modeling to generate a digital twin that we can throw optimal control theory at to make a prediction of what the therapeutic regimen should be for answering these two questions here on the screen there. And then the last bit here, we're almost done, is to try to update our interventions using data assimilation, right? Because you're not just, patients just don't get one therapy and then it's over with, right? There's multiple therapies, multiple treatment time points, and you're going to want to update your predictions. And so fortunately, again, the weather modeling people developed methods to do this. So again, super fast intro to data assimilation. It allows newly available data to be integrating with predictions of a mathematical model to refine the characterization of a system that is dynamically changing in space or time. And there's lots of ways to do this. This is a really famous example of this technique. So this was Hurricane Sandy, which was, I believe, 2012. And so this was a big deal, right? This was a really big deal also from the point of view of weather forecasting, because 72 hours before this hurricane made landfall, the prediction of where the hurricane eye was going to hit was correct within 72 miles. And so that was an enormous leap forward in predictive accuracy. And you can imagine that that would be extremely important if you're trying to evacuate 15 million people from the Northeast corridor here. And so these are different, all these little lines correspond to different predictions made at different times. And so the initial ones here, the purple one and the lime green one right here, or the teal one's easy to see. It kind of goes off out into the Atlantic right here, indicating it may not be as a huge problem. But as more data became available, then these predictions got more and more refined until, then within, 4 or 5 days here, you're making pretty accurate predictions of that it was going to change course and go right into the Northeast corridor right there. And so the data is being assimilated at each time point, at each prediction time point. Of course, they have way more data than we do, but that's life. All right. All right, so we've started to apply this technique to predicting growth and response of cells to radiation. And so what we did here was kind of a mixture, a poor man's mixture of mechanism-based modeling with a more big data, in quotes, kind of thing. We wanted to predict from the earliest time point. So we took time-resolved images of cells growing in dishes that were receiving radiation therapy. And so we have this, lots of people have these devices where you can put dishes in these incubators that live, or inside of, you're putting the cells inside these machines that have microscopes on that live inside an incubator. And so you can leave them there for weeks and you get lots and lots of data with lots of different interventions going on. You put 96 well plate in there. Even if you have 8 replicates, you got 12 different interventions that you're looking at one time. And so we looked at a whole host of different radiation interventions and hundreds of data points on hundreds of samples. And so we took 75% of them to figure out, to train our model. And then the 25% that we held back, we wanted to predict from time point 0 using the baseline that we got from our population train thing to make our first prediction. And then as more and more data came available for the individual time course, we weighted our predictions on the model calibration over here, more and more than less over here on the population. So population, individual, predict from time point 0 when there's no information beyond initial conditions for the individual, and you're doing that prediction based on the population. And then as you go further and further on the individual, you include more and more of their information. So you fit a training group globally to get some set of parameters from the population. That set of parameters called X. So you can make a prediction for your new person right here. And now you've had this time that they've been growing, and so you're making your measurements there, your time resolved microscopy data up to time point 1. And these images they're acquired like once every 3 hours or whatever. And so now you have 8 time points to calibrate your model, which gives you a set of parameters, but you only have 8 time points for these things. And so you weight those model parameters by your population, and then you predict forward going to the future, and then this repeats itself. And so, by the time you've reached half of the time course of your experiment, you're looking just at the individual, and you're not having any weights from the population. So if you just start from the global parameter, and that's this green curve right here, then your prediction at the end of your study, 300 hours later, 2 weeks later, whatever that is, you're far off from where you actually are. But if you keep making these model updates, then you're doing pretty good, and it looks like it's about time to make another model update as this prediction is going off, and the data seems to be not increasing at that much rate there. So at early time points, you're weighted a lot by the individual, but then by the time you get to right about here, you're looking only at the individual. We've done preliminary results on this for GBM, and those results are pending. We just had a meeting on that right before there. It is a difficult problem. I'll just say that. And here's more examples for what's going on from, in some cases, the prediction you have from time point 0 is pretty good, and your big data approach works. But in lots of times, your prediction that you get just from the big data at the beginning is not working out very well at all. And so you really need to have model updates with your individual data. So we're at the point where I summarize, but no, I'm going to put one slide in here to tell you about some other things that are going on in the lab, just so you think it's not just all in breast cancer. We're applying these techniques to brain, prostate, and just recently started cervical cancer. We're trying to do proper mechanism-based modeling with neural networks so you can do predictions from before there's any intervention with the individual patient, just like we were talking about for those cells. We're trying to predict the distribution and treatment effects of these radiolabeled nanoliposomes. They're giving really high doses of radiation in GBM. In the preclinical setting, we're trying to build these mechanism-based ODEs and linking them with a time-resolved microscopy, similar to what we just talked about for various interventions, trying to incorporate hypoxia measurements into the radiation treatment models for obvious reasons. And then we've started modeling the effects of immunotherapy. We're probably behind the curve on this, but it's a difficult thing. And then we have lots of theoretical projects, working on model selection, identifiability, and so forth. All right, so the summary: optimizing outcome relies on considering each patient's unique characteristics. This can be achieved through the mechanism-based modeling, the digital twins that you can throw optimal control theory at, and then you have to update your predictions as more data becomes available. So the point, and this is the last slide here, when he left Harvard, it was clear to him that if you wanted to design something for an individual human being, the average was completely useless. So if you want to design something that is useful for an individual patient, you got to rely on their characteristics. And there's now a suite of mathematical techniques that exist and are practical for doing this. I hope I've convinced you of this. And this effort to mathematize oncology, it's kind of inevitable, right? Once we mathematize a subject, if you look at the history of science, we get pretty good at it pretty quickly. It's already happening in cardiology. Oncology is probably 10 years behind, but this type of thing is going to happen. And these are, of course, the people who do all the work. I've highlighted the work of the people pointed out here. I, of course, just sit in the corner and worry about how to pay for it all. And thank you very much for your time and attention.
Thank you, Tom, for a very nice overview and enlightening talk, and very entertaining too. You did not disappoint. There are a few questions in the chat, and I also have a few questions. Let me start with the chat. So Harrison Kim is asking, could you explain how to incorporate immune response? And I guess the more general question is, how do you integrate the tumor microenvironment?
Yeah, so these are really complicated questions. When we started this out, we went to the whiteboard and we listed everything we could measure, and then we went to build models where we incorporate these things. So we've tried to take a very parsimonious modeling approach. When we feel like we need a new measurement or a new thing that needs to be accounted for, then we do it. And we've gotten to the point where we have to start thinking about the immunotherapy. And the short answer is we don't have good ways to incorporate the effects of immunotherapy in these models. They are going to be implicitly incorporated in there because that proliferation term, that logistic term that has the K out there, that's a net proliferation. So it's the net result of the fighting between the tumors proliferating and them getting hit with whatever treatment. So it's buried in there implicitly. I understand that's not a very satisfactory thing. What you really want to do is probably add another term on the right-hand side there that's going to have death due to immune cell activation or whatever. But then that would give you another equation that described how the immune cells are getting there. And every time you write down another equation, there's things on the right-hand side that you either have to measure or make assumptions about. So it's, we've poked around with trying to use some of these zirconium PET tracers that are going to give you hints on where some of these immunotherapies are landing, and maybe use that information to try to tell us how the immune cells should be either activated and or migrating to the tumor site to do their stuff. But this is really just at the stage of whiteboard or writing down families of models that might work, but we really haven't done much calibrating on that front. So it's an open area of investigation. Everybody's struggling with it. If somebody wants to work with us on that, I would be thrilled, because that's hard.
Let me go to the next question. Are you proposing combination methods such as PINs? I'm not sure what PINs is. Do you know what PINs is?
Yes, that's physics informed neural networks. Right. Yeah. So that I have to answer this carefully, because as far as I know, I'm the only person who hasn't drunk the big data AI Kool-Aid, and so I always am stepping on people's toes and irritating them and stuff. So I that there's two ends of the computational oncology spectrum. There's the AI big data stuff, and then there's the more mechanism-based stuff. And of course, this is where I live over here. There is, of course, truth in between, and there are certainly examples where they need to come together. And so we're sort of dipping our toes into that with trying to have if you have a large enough patient set, you can calibrate your biology-based or mechanism-based or physics-based model to that data, and have that as a training data set. So when the new patient comes along and you have their initial conditions, you can map through a neural network, for example, from your training data set to this individual to initialize her model from time point 1 to run forward. And then as you get more data from her, you can update your model to have more specific patient-specific predictions. We had our first effort at that was an abstract at this past AACR, but there's a lot of work to be done there. So am I advocating for it? I think it's definitely a way forward, but I really feel like you have to have these mechanism-based models that make use of the individual patient characteristics, because humans are largely outbred. We're so different from each other that the heterogeneity is what kills us in this disease, right? It's the disease that works for patient X is not going to work for patient Y. You really have to calibrate your models with their individual data, and you really have to update your model predictions with their individual. I think there's only so much you can learn about the individual from the avalanche of data. So only so much you can learn about the snowflake from the avalanche.
Yeah, I agree with you on that as well, to some extent. I think
To some
there's a lot.
extent, yeah.
I think maybe that's calls for maybe a panel discussion about where the future holds for AI, machine learning, and integration with more mechanistic models. I think there's room for both.
Yeah,
We're getting ahead of ourselves with the
right.
big data ideas. The next question is from David Buckley. How do you deal with complex treatment time courses when you only have imaging data at very limited number of time points, which likely will not be optimal?
Right. Yeah. That's for sure. We are data starved in that direct. So what we do is we use our DCE analysis to estimate how the drugs are being delivered, how they're initially being delivered. And I understand that is probably making a lot of you want to punch me in the throat. What we found is for the broad spectrum chemotherapies, the small molecules, the gadolinium-based chelates, they're roughly the same size, and they do a pretty good job of estimating how the drug is going to be distributed, and the predictions are pretty accurate for how the patients are going to respond. So what we do is we use the DCE stuff to give us an estimate of how the drug is being delivered. And then we have to go to the clinical trial literature. Phase 1 trials, of course, have all the pharmacokinetic data in for how a drug stuck in the bloodstream for the patient. All phase 1 trials take blood samples from the patients after the drugs are administered, so they get these sort of bulk population time courses for the washout. And so we use that to estimate the drug concentration change in the blood over time, and then you use your DCE data to estimate how the drug is being extracted. And I understand, like that's not the best approximation. So that's why I was very careful to say, to first order, these are approximations of how the drugs are being delivered. And it's proving to be not that bad. For the drugs that, the monoclonal antibodies, that is our estimations of how the drugs are being delivered is not as well, and our predictive accuracy is not as good there as it is for the small molecules.
Maybe there are a few more questions, but let me follow up on that idea, on that topic, the drug treatments. So from your mathematical formulation, you're basically just capturing the concentration of the drug in tumor. There's no mechanism of how the drug is impacting the tumor.
Oh,
So,
right. So in the model, the mechanism is buried. The mechanism of action for the drug is buried in the efficacy parameter that's in front. That's, if you can remember, it's the last term on the equation on the right-hand side is the treatment term. I had a little box that said treatment. And
right.
there's the concentration inside the summation, and there's an alpha up there, the alpha sub n. That's the efficacy of the nth drug, and so that alpha has to be calibrated with patient data. So the idea is you have the baseline, and then early in the course of therapy, and then you can calibrate that model to give you that parameter to give you an estimate of how efficacious that drug is. But the mechanism is just buried in it; it's not explicitly incorporated.
So, but what do you use to calibrate that alpha per patient?
Yeah. So if I go back to the model, it is coming up rapidly. It is. We're almost there. Just another. You're listening to the Tom Network, WTOM. All right, here we go. Right here. So this is our treatment term. So the things we measure that we're estimating, the ends are estimated from our cell density maps for diffusion data. The theta is measured on the voxel size. The drug here concentration is estimated from the DCE data and then washed out according to phase 1 clinical trial pharmacokinetic data. But then so everything else needs to be calibrated for. That means we calibrate for D globally, alpha globally, and then K locally. So that's the thing that's fit during that calibration step.
I see. Okay.
Yeah.
Let me ask more questions here. I guess somebody's asking about photothermal therapy models in mice, and I'm guessing how this could be applied in photothermal application.
So we've talked with people about that. We've never gotten our hands on any data for that, but that's the power of this whole mathematical modeling nonsense. This treatment term, it's just a module. So like when we go to radiation therapy, this treatment term is swapped out for the equation we want to use to describe for radiation therapy. So in principle, we would swap this out with an appropriate model describing thermal ablation, for example. What's the rate at which you're destroying tumor cells with phototherapy, for example. So we haven't done that, but in principle, it's a whole project. We'd have to construct the model, do sensitivity analysis, all the stuff we normally do that's behind the scenes that nobody cares about. And then we'd have to marry it to the data. But in principle, these treatment terms in this box here, that is just swapped out with whatever the intervention is that you happen to be looking at.
Right. Let me ask another question. So as I'm sure you know, this whole concept of radiomics and so forth, how do you integrate radiomics in this concept of this mechanistic model, and to what extent can you combine both?
Yeah, we haven't done a whole lot with radiomics. We made one or two efforts on this where we were we did the radiomic thing to go on the great Bob Gillies idea of these things are forming habitats, right? So we built this system of ODE, ordinary differential equations, to track how the cells were moving between habitats, not actually physically moving, how their habitat was changing. And so we did do the small scale radiomics to come up with different habitats that were, high perfusion, high cellularity, or low perfusion, low cellularity, and all the combinations of that. So it was a system of 3 ODEs that described how the cells were, or the regions were migrating between that. So we built an ODE system describing how the different habitats were changing over time. And could that be used to predict if a tumor was going to become more or less responsive? This was radiation therapy in a rat model of glioma. So we've done that. We're trying to now expand that to include hypoxia in the habitats and then apply it to patient data. It's still pretty early in that. Maybe something at ISMRM next year, but it's still pretty early on that. So that's how we've tried to combine the mechanism-based modeling with the radiomic/habitat stuff.
I'll ask more questions then. As you mentioned, when we apply machine learning and data science techniques on the population scale, there's scarcity of data. Have you thought about implementing these strategies to stimulate patients to generate data to use for machine learning and AI type applications?
We've thought about it superficially. We've talked about it with others kind of superficially, but we've never done anything like that. We are, there's only so many hours of the day and there's only so many dollars in the bank account for our lab. So we haven't done that. In principle, it should be a tractable thing to do. So if people are interested, please shoot me a note. We don't have the people power to do that. So we can't do that. But if people are interested, then you could imagine using some of these models as an engine to do just exactly what the person suggested.
Right. I don't see any other questions. Let me see Q&A chat. No, I think we're good. It was very
Okay.
thank you for a very nice and informative talk, Tom.
Well, thanks
I
for the
think we
opportunity.
really enjoyed it.
Thanks so much for the opportunity. I really appreciate it. Thank you.
All right, I think we can end I guess the conference and the Zoom.
All right.
Thank
Take
you
care,
all.
everybody. Thank you for your time. Thank you.